Welcome, everybody, to Chapter 6 and finally, Week 7 of the course. So, microbial nutrition and growth. One of my favorite things about this topic is that it comes back down to evolution and natural selection. You've heard me say that before. But when we're thinking about diseases and we're thinking about treatment, we're thinking about ways to prevent disease, then it's going to come down to some pretty basic kinds of things. Tolerance ranges, where these things like to live. We call it cardinal temperatures. So that's one of those. What nutrients, what vital nutrients do they need? And if we rob them of those nutrients, do they die? So it's going to go on and on and on. But if you can understand really the ecology and really kind of the environmental factors that affect bacteria and viruses, then we can better learn how to prevent and treat disease. So kind of think about that as we're going through this week's topic. All right. Microbial nutrition. Honestly, when you look at it just at face value, it looks very similar to human nutrition. So we have essential nutrients, essentially, organisms have to have. It's something that has to be provided to them. Macronutrients, macro means large. So these are the nutrients that we need in larger quantities. And these are the ones that, are making up our macromolecules. So carbon, hydrogen, oxygen, nitrogen, phosphorus, another form phosphate, and then sulfur. So not that much different than what you need. So these are going to be the basic building blocks of macromolecules. They're going to be the basic building blocks of the building of plasma membranes and cell walls and the lipopolysaccharides and so on. So kind of keeping in mind that there's a reason why microorganisms require nutrients, and that's so that they can build all the things that they need. Now, there are what we also call micronutrients, and humans have those as well. So these are the nutrients that we need in much smaller quantities, but we need them. So manganese, nickel, zinc, not that different than humans. So when we take a look at, this is the chemical composition of E. coli, and what they have done is essentially dried out the E. coli so we can calculate percentages based on dry weight. Then you can see there's that carbon, oxygen, nitrogen, hydrogen, phosphorus, and sulfur. So those are the top elements in that chart. So these are our macronutrients. They can also see on the left-hand side organic compounds. So these are our macromolecules. So essentially, we're starting to get an idea that if 50% of the dry weight of E. coli is proteins, then we're going to need those particular elements in a higher concentration. So kind of a way to look back and forth between these two sides of the table. The inorganic compounds are ones that are other than the carbon-hydrogen compounds. All right, so some of you have heard this maybe in a prior class, this idea of where does your carbon come from? All of us humans, our carbon comes from being heterotrophs. Hetero means different. Troph means the feeding level that we eat at when you think about the food chain. So heterotrophs, we require the ingestion of our nutrients from other organisms, right? So whether you eat plants, so whether you're a vegetarian or whether you're omnivore, carnivore, we have to consume other organisms to get our carbon. But our photosynthetic creatures, some of my favorite things, of course, plants and algae, either microscopic algae or things like kelp, these things are autotrophs. So auto means self, troph means feeding. So these organisms have the necessary machinery to use inorganic carbon dioxide as its carbon source, right? So plants, for example, have pores underneath their leaves, and these are called stomata. And CO2 comes into the plant right there at the leaf, and oxygen leaves right there at that same pore, the stomata. So autotrophs are interesting because they technically are not nutritionally dependent on other living things. Now, there's always that one exception. So you may say, hey, well, what about the Venus flytrap? Or what about that white plant? It used to be called Indian's pipe. It has a new name. I don't remember it. But that plant is all white. So what do you do about, the Venus flytrap needing to eat flies, are a carnivorous plant, so to speak? And then how can you possibly have a white plant? Well, there are exceptions. So the Venus flytrap is eating or capturing flies to get that essential limiting nutrient we call nitrogen. And when we think about a plant that's white, does not have the pigment it needs to do photosynthesis, so it's likely feeding off another plant, and it's usually feeding off the roots of a tree. So it's getting all of its nutrients by stealing it. So there's always, some exceptions. Our last category here that we're going to talk about are the saprobes. So these are, it says, free living organisms that essentially are consuming dead organic matter in the form of what we call detritus. So if you go out into the woods, you get a really good idea of detritus. You've got leaves, you've got like dead, broken up parts of pill bugs and grasshoppers and twigs and maybe some decaying fungi and so on. So when we think about saprobes, they are essentially the decomposers of plant litter, animal matter, so leftover parts like tendons and ligaments and of course fecal material. And then saprobes are feeding off of dead microorganisms. So gotta love saprobes. This is why I'm not too worried about the leaves in my yard, is that they will break down and the nutrients will be recycled, and I won't have to spend time or energy dealing too much with my leaves. And saprobes are very important to recycling organic nutrients. So then we're thinking about the nutrients in the environment being available to autotrophs or being available to heterotrophs by eating autotrophs. That's where it's coming from, these recycled nutrients. All right. So we want to think about in a little more detail, why might microorganisms need these particular essential nutrients. So I already mentioned it a little bit before, but if you like it kind of detailed out, it's here. So carbon is going to make up our macromolecules. So proteins, lipids, nucleic acids, carbohydrates. Hydrogen, also super important to those macromolecules, but let's be a little more specific. Very important to maintaining our microbial pH and in helping in production of energy and cellular respiration. So you might have remembered I was talking about the electron transport system last time. So hydrogen is very important to that. Oxygen, so structural and enzymatic functions. So we talked about oxygen, being an electron acceptor. So oxygen, as you're going to see after this week's lectures, is super important because organisms either require it and know how to break down the toxic effects of it, don't use it at all and don't break it down, or use it and can break it down. I think I just covered them all. So essentially it's super important because oxygen, free radicals of oxygen or superoxides can be very dangerous in cells. So come back to that. Nitrogen. We're thinking about nitrogen. I'm always thinking about amino acids, and amino acids make proteins. We also know that nitrogen is important with the making of DNA, RNA, and ATP. So those are our nucleic acids. All right, phosphate or phosphorus, important to protein synthesis. Membrane function, so when we're talking about those lipopolysaccharides, and yeah, pretty much, and some other parts of the cell membrane or plasma membrane, it's important to have phosphate there. And of course, it plays a role in ATP. Remember how we went from ADP, adding a high energy phosphate bond, to making an ATP. And then lastly, you might remember there was an NAD and NADH. So phosphate is important to that coenzyme. Okay, lastly, we have sulfur. important to the manufacturing of vitamins and amino acids. So there's a couple of different terms that we need to introduce. I'm sure you've heard these before: diffusion and osmosis. So when we think about this, the reason why we need to think about this when we talk about bacterial cells is it helps us understand how materials are getting moved from inside the bacterial cell to outside. So in diffusion, we're thinking about molecules moving from a region of high concentration to low concentration until they reach equilibrium. So you can see here in the figure, the far left, there's a blue dropper, and a person is dropping, a couple of drops of that blue dye into a beaker of water. Now you don't have to stir it or anything, and you can just watch it, and you'll see that that blue dye essentially is, for lack of a better term right now, mingling and bouncing against the water molecules until essentially the blue dye is in equilibrium with the water. So now you can see on the far right hand side that that blue dye looks pretty much consistently the same color throughout the beaker. So anytime we have the movement of molecules from a high concentration to low concentration, same thing can happen if you spray perfume, like you spray some perfume or cologne or air freshener before you leave the house. And then, someone upstairs goes, "Did you spray that stuff again?" Well, because the smell or those air freshener molecules or perfume molecules then are essentially dispersing by, mingling with the air molecules in your home until it goes from the high region of concentration to the lower region, which might be upstairs in a bedroom. So osmosis is diffusion of water, and we usually describe it as the diffusion of water. Okay, so think in high concentration to low concentration, but through a selectively or semi-permeable membrane. So let's take a look at this for our last topic in this first video. If you take a look at the beaker, you're going to see that essentially there looks like there's a pipette with some kind of balloon on it, and that balloon is this special semi-permeable membrane. So that membrane is only going to let molecules in of a certain size. So the solute that is in that balloon membrane is too big to leave, okay? But we have water, of course, both outside the selectively permeable membrane and inside. So what do you think is going to happen? If you have a high concentration of solute inside that semi-permeable membrane, then you're going to have less water. And on the outside of the semi-permeable membrane, you're going to have more water molecules than you are, of course, the solute. So the water is going to move from the region of high concentration, which is the water outside in the beaker, and it moves through that semi-permeable membrane, bringing that solution into equilibrium. So you may ask yourself, well, where does that extra water go? And that's why there's that glass tube or pipette. Okay, so it kind of gives you an idea what's going on with osmosis. And so we know that water is super important in cells, and so we have to be able to move it to where it needs to go. This will have implications for nursing when people are coming in and are dehydrated or need electrolytes of some sort. We're thinking about giving people fluids, no matter what it is, if it's albumin or, like standard normal saline, or if it's, like a higher or lower amount. It's usually what we're trying to cause some fluid shifts in the body, and we have to be very, very, very careful about it. So understand osmosis so when you get to those topics in like a nursing class that you're not feeling overwhelmed. In this figure here, on the left-hand side, so what this tells us, isotonic solution, it tells us that the solution inside the cell is at equilibrium or has the same amount of solute concentration as outside the cell. So the net movement is essentially zero because it's in equilibrium. Now, if we take a cell and we put it into a solution that has a low amount of solutes in it, And here they're saying the hypotonic solution is